De comenzar mencionaba que me gustaría agradecer al TEC y a la Academia Mexicana de Ciencias por el apoyo para que este evento se hiciera realidad. Asimismo agradecerle a Enrique Luna, a Irán, a Paola y a todo el staff del Centro del Agua por hacer su trabajo. Sin ellos pues es difícil de que este tipo de eventos se organice. Les voy a agradecer a ustedes por estar aquí, por hacerle preguntas al profesor. And especially also I would like to thank Professor Logan to give us 2 days or 4 days of very precious of his
4 days and 2 days.
2 days to be here and 2 days of traveling, yeah? So that's why thank you very much again. And I hope could be one of several times that you could be here also. Sharing your experience, your expertise, and especially I think these events are very important for the students, yeah? Okay.
Okay.
Again, the hall is yours, and the students also are yours.
Okay, thank you. Okay, well, good morning, and thanks for braving what I understand is very, very cold weather. Not to me, but I guess for you. So I see lots of scarves out this morning. Winter coats, yeah, see? It's okay. It's fine. So yesterday we talked a lot about microbial fuel cells and microorganisms, the architecture, and scaling these systems up. And today I'm going to shift a little bit to microbial electrolysis cells and talk about what happens when you run these reactors now in oxygen-free environments. And I modified the title a little bit here just to say biohydrogen production, because basically at the cathode, we're not running oxygen reduction anymore, we're running reduction of some other chemical. And so we're going to talk a little bit about what happens in these systems, what makes them now depart from being like microbial fuel cells or stay a little bit similar. I won't talk so much about scaling up the systems, just a little bit about that. And then I'll move into other products other than hydrogen in these systems. And so just to refresh our memory, a microbial electrolysis cell typically has bacteria on the anode. They oxidize organic matter, release electrons into the circuit, and unless a little extra voltage is added through some sort of power source, nothing happens. But with a little bit of extra voltage, you can get hydrogen evolution at the cathode, and that can be harvested as a way to convert organic matter into hydrogen from things which are thermodynamically not favorable. So for example, acetate is not favorable for hydrogen production. It's usually a dead-end product of fermentation. If there are other microbes on the cathode, we can drive other reactions, either directly from electrons derived from the cathode or from hydrogen evolved in the cathode. And for example, we can produce methane electrochemically in this system as well as hydrogen. Of course, you could also split water and make hydrogen, or split water and do the same thing with the methanogens. So it's also a way to turn, for example, solar energy into hydrogen and methane and perhaps more complex organic products. So this is taken from a figure we did a few years ago, just looking at saying, you know, what's the product value of some things? Well, it turns out electricity is actually not very valuable when you look at its electron equivalent value. Hydrogen's more valuable. Methane, again, not so much, but you can then think about driving some organic products which then become greater value. So this has created a community which looks at what's called microbial electrosynthesis, which is to use these cathode-derived electrons to actually produce organic substrates by using CO2 as the carbon source. One of the nice things about using MECs is also to consider not just wastewater treatment, but if we could make these technologies inexpensive and effective, we could also look at harvesting other sorts of fuels for these systems. And one that's been of interest to me is the fact that in the US they've estimated they could produce about 1.3 billion tons of cellulose without impacting food production. Now, 10 to the 11th kilograms per year would be derived from that amount of cellulose, stoichiometrically, and that's 12 moles of hydrogen per mole of hexose equivalent. It's also of interest to me that in the US, if we were to change to hydrogen as an energy source for light-duty vehicles, that's cars and light trucks and that sort of thing, it was estimated we need about 10 to the 11th kilograms per year of hydrogen in that transition, ultimate transition. So, you know, it's foreseeable that we could run all the vehicles in the US just on hydrogen from waste biomass or biomass produced. So this is a very interesting prospect. Now, if you look at converting cellulose into hydrogen, you can write out stoichiometrically cellulose going to hydrogen, but biologically, if you do it, it dead ends at hydrogen, and the best product would be acetate. So the most hydrogen you could get would be 4 moles of hydrogen, but you would also end up with 2 moles of acetate. And so the maximum conversion microbiologically is 4 moles of hydrogen per mole of hexose. That means you're throwing away 8 moles equivalent because you don't know what to do with it. And that was what was called the fermentation barrier. But this barrier can be surpassed by using microbial electrolysis cells because now that acetate becomes an optimal substrate for hydrogen evolution in a microbial electrolysis cell. In fact, I've been working for a few years with the National Renewable Energy Laboratory in Colorado, where they have been working on dark fermentation processes to convert lignocellulose and really cellulose into hydrogen by a fermentation process, releasing primarily acetic, but also other volatile fatty acids and some ethanol. And then the idea is we use our system to convert that, and our record for conversion is about 11.5 moles of hydrogen per mole of hexose equivalent, which is, you know, we think pretty remarkable. The energy yields are also pretty interesting here because we can beat thermodynamics. That is, we can create more energy than we put in. Okay, I didn't really say that. Nobody would believe that, right? But if you just look at the electrical energy that you're putting in to make this hydrogen, you can actually get 2 to 4 times more, 200 to 400% more hydrogen out than the electrical energy you put in. And that's because you're neglecting the substrate energy, and of course, if you include that, you know, you're not going to get above 100%. But up to maybe 90%, but of course the rates would be very slow. As you increase the rates, you become less efficient and less energy efficient. So it's a balance between how much electricity you want to put in and the rates of hydrogen production that you want to get out. So with that backdrop, what I'd like to talk to you today about is, in this lecture, are 3 basic topics. 1, what happens to the microbes in these microbial electrolysis cells? How is that the same, or how is that different than fuel cells? The big difference being that oxygen can leak into the reactor when you have a microbial fuel cell, but there's no oxygen in the electrolysis cell. A little bit about some of the reactors that we've used, and then talk about the microbes that are involved on cathodic reactions, primarily methanogens, but also some information on microbial electrosynthesis. And there's a lot of interest in microbial electrosynthesis, but my question is it the best way to run a reactor using the electrodes to make the hydrogen, or are we better off just splitting water and using that hydrogen? And I'll get back to that near the end of the talk. So first, the microbiology, these electroactive biofilms. We study these in the lab, and when you're doing microbial electrolysis cells, a lot of times you want to set potentials of electrodes, and so now you need to have a potentiostat. This is a 16-channel potentiostat made by Biologic, which is absolutely fantastic. Potentiostat, it's the best one we've ever used, and we've used several of them. 16 channels at about, I don't know, $3,000 or $4,000 a channel. It gets really expensive really fast. So these instruments run $50,000 or $60,000 a piece. You don't have to have one of these to do this research, but it sure helps for some of the electrochemistry. The other thing is, because you're producing gases from these reactors, we also often just collect these and say gas bags, and then we can analyze the concentrations later on. Some people use water displacement. The difficulty with that is sometimes you can lose the gas through solubilization, but most of these gases produced are not that soluble. So let's start with the anode before we go to the microbes on the cathode. Yesterday, we talked about analyzing microbes from wastewater treatment plants versus bogs, and we said that when we looked at the reactors from these different sources, mostly we got delta proteobacteria on the anode, and most of those delta proteobacteria were indicated to be Geobacter, and most similar to Geobacter sulfur-reducens based on 16S RNA sequencing. In an MEC, we add voltage or we set potentials, unlike microbial fuel cells where you're usually not setting potentials, but in one set of experiments, we set potentials in microbial fuel cells and just drove the cathode at whatever potential it needed to be to run the reaction. And so this is the different potentials that we set here indicated. And what you see is that as we set these different potentials, we drove more current, we produced more biomass. But the point is that we didn't really impact the microbial community. So even over all these ranges of different potentials that we might choose to set the anode at, we almost always got Geobacter. So at least from the standpoint of setting of an electrode, the actual potential of the electrode itself, it didn't seem to change the community that evolved. We've also looked at what happens when we have complex substrates. This is a potato wastewater where we were running these anodes in microbial fuel cell mode with oxygen reduction, and then we switched them over to MECs. We took away the oxygen cathode, we put on a cathode where we evolved hydrogen, and what you can see is it looked like there was some shift in the community, but not much. We still ended up with mostly Geobacter. Maybe metalloreducens was typing out more than sulfur-reducens, and then about half of the other community was probably involved in the degradation of the starch and complex organics into volatile fatty acids. We also looked at winery and domestic wastewaters, and again, these are systems that were shifted into MEC mode. And again, you can see here a pretty big abundance of sulfur-reducens. So the take-home message from all this is that between MFCs and MECs, we don't see a real big difference in the predominance of Geobacter. The complexity of the wastewater will impact the total community because it's much more diverse now than when we feed it acetate, but it's still going to end up being Geobacter on the anode that produces the products. Now, what's interesting to me is that when we actually went in and isolated one of these Geobacter, it typed out as 100% similar to Geobacter sulfur-reducens. But when you actually looked at its functionality, not its 16S gene, but its functionality, it was quite different. And in fact, we named it a different species, Geobacter anodoreducens, which was isolated. In this case, it came from an MFC formate. It had much different DNA hybridizations. It was much more tolerant of salt, which is very important in our systems because they have a lot of phosphate buffer, and we want to have salt to increase conductivity. It grows very well in 200 millimolar phosphate buffer, but Geobacter sulfur-reducens PCA strain does not. And maybe most importantly, it cannot use fumarate as an electron acceptor, which is sort of one of the hallmarks of sulfur-reducens PCA. So even though your 16S RNA is saying geobacter, 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 it may not be the geobacter that is in the, in the typical storage locker, shall we say. So the student then, postdoc that isolated this, compared the performance of this inodoreducens to some mixed cultures and to also sulfur-reducens. And basically, these sulfur-reducens can form thick biofilms. They tend to be pretty homogeneous. And she became interested in what happens. This is Dan's son. She's now at Zhejiang University in China. She became interested in what happens in terms of the viability of these biofilms over time. And this was, people had been talking about geobacter biofilms only being very thin, 10, 20, 30 microns, but this biofilm was quite robust at 100 microns when mature. So the question was, okay, if we have this biofilm as it gets thicker and thicker, do we get substrate limit, do we get electron acceptor, that is the ones that are far from the electrode, do they not have access to this electrode, but they have access to substrate out here? Or is it the other way around, that the ones that are on the electrode get outcompeted by the ones up here that have all the food, right? So if the food's coming in here, who gets it? But whoever gets it also needs to have the electrode. So is it homogeneous? And she used two stains. The live cells are green, dead cells, and it doesn't mean they're actually dead. It means that their membrane is compromised enough for the stain to leak into the cell. So it's not truly, we don't truly know it's dead. It's an indicator, it suggests that it might be dead. So she grew up these biofilms. She used these little mini MECs, which I'll talk about a lot as we go on. I brought one. It's really tiny. This is an example of a very small reactor. You can use the same concept to build bigger ones. But basically, it is literally two electrodes stuck in a reactor. We use graphite block for the anode and a stainless steel screen for the cathode. And we add voltage using a power source. And because she had graphite blocks, she was able to do very good confocal microscopy and sectioning on these biofilms. And so this is what she sees over time. Each cycle is when you inoculate the reactor, then it uses up the substrate, so you pour out the substrate, the media, add fresh media. So every fresh media is a cycle. And what you could see were 4 different phases. The first phase was increased current due to development of the biofilm, high current density, and then a characteristically drop in current over time, and then some sort of new steady but lower current density than she had initially seen in the system.
The system got a relaxation time at what time? Let's say in the order of
Oh, no. So this is actually the maximum current from one cycle. So we feed it, and how much current did we get in that cycle? Well, we got about 1 amp per meter squared. And then the next time we fed it, so this is the maximum current in the 5th cycle, the 12th cycle, and the 30th cycle, and then 30 to 50 cycles. So 30 to 50 cycles spanning, these typically go 1 or 2 days, so about, could be anywhere from a couple months to 3 or 4 months, something like that. The other one up here is a mixed culture. And so that shows you not so much that characteristic up and down. It pretty much tends to come up and level out close, but sometimes maybe a little bit higher, but maybe not statistically significantly different. You can compare them over here, and you can see this mixed culture coming up, and again, this sort of plateau in current density. So what does this look like in terms of the structure of the biofilm? This is the biofilm C1, that's initially after 5 cycles. And you can see this little kind of red layer developing down there, and then a green layer on top. And this becomes far more pronounced as it gets thicker, and you can see this now thicker red layer. So what this implies is that the biofilm near the electrode, which is down here, the biofilm near the electrode is either dead or starving or membrane compromised, and that the cells on the surface tend to be the ones that are most active, maybe scavenging away that substrate before the ones in the bottom can use it, or maybe they just die off, we don't know. Mixed culture biofilms, of course, much greater diversity of microbes here. But again, while it's a little bit more spread out, you can still see this sort of layer-like system developing. So even though there are other microbes present, it doesn't seem to really alter that situation. So this is the bottom line was a dead red inner core and a green outer core alive. This was not expected, actually. We thought it would be exactly the opposite. We thought the biofilm near the electrode would be the viable one, and the one on top would be kind of pushed off and away. In fact, I had given lectures where I said, this is probably what's going on. When people would say, what do you think is happening? I'd say, oh, I'm pretty sure. So, but the data do not support what I was saying. This is what our data support. And there's other data in the literature that actually says the opposite. So this is really kind of a very still open question about what the structure looks like. But we did some experiments to really say, you know, what possible artifacts could be leading to this? Was it the use of this inodoreducens rather than sulfur reducens? Well, no, we did it with sulfur reducens, and you can see the same sort of red inner layer near the electrode. Okay, so it wasn't the specific microbe we used. Was it due to the use of this single chamber MEC, because hydrogen is coming off the cathode, and Geobacter can use hydrogen. Some strains can use hydrogen. And so they're sort of cycling that hydrogen. So maybe something strange is happening because of that hydrogen. But we did the same experiments in a two-chamber where the hydrogen does not contaminate the anode. And again, we saw the same sort of evolution of a red inner layer and a green, or turns a little bit yellow, outer layer. Was it the current density, and how did the current density possibly play into this? And of all these, this is really the most confusing one to me, because as you increase the voltage, you think you would increase the rate of consumption by the microbes near the solution. And so the dead part, I would expect from sort of a chemical engineering perspective and diffusion and mass transfer, I'd expect more of the more dead cells and, you know, less green cells. That is, they're working really hard. But what happens is opposite to what I would predict. So, you know, don't ask me to predict the future. It actually, at the higher current, it actually turned more uniformly green, and we don't know why that is. So there's lots we don't understand about how this community forms, and how we know that Geobacter use conductive pili, and that these pili remain conductive even if the cells die off, because they're just conductive proteins with cytochromes embedded along the pili. But we don't know how other microbes use that or take advantage of that, and how the biofilm itself evolves. And, you know, part of our thinking was actually that as it started growing faster, it actually, this totally viable biofilm displaced some of those dead cells. But we have no scientific evidence for that. Yeah?
In order to elucidate what is the possible reason why you can see that phenomenon, I have a question. I know that first it's important to get rid of the possibility that this system is dominated by concentration.
Uh-huh.
So somehow I see that the experiment was done in this little glass.
Right.
So if you put, for example, a magnetic bar, even though that it's not the best as a magnetic silicon,
Right.
somehow can give an idea just by force convection if you have an influence of that or not.
Yeah.
Can you do this kind of experiment?
No, we have not done it, and you could do it in larger reactors. It's a little tough to get a tiny stir bar in that little one. You could do it in larger reactors. Some people have grown these on rotating disc electrodes under, you know, reasonable shear. The problem is that the higher the shear, the more likely it is you're going to pull the biofilm off the surface. these are not well held on. And there are pictures of people that take like tweezers and just kind of lift this thing off like a piece of wet paper off a surface. But it's certainly worth doing. I mean, we have not done that. So, this is this high current, low current situation is a real question. We don't understand it. There's obviously something going on in terms of metabolic status, pH, community interactions, quorum sensing, I don't know, although I don't believe that this strain is known to have any quorum sensing signals. It may have receptors for that, but it's not known to produce these quorum sensing chemicals. What are the homolactoserines or something like that? So that's the microbiology part of this. You'll see these biofilms dominated by Geobacter, various species, but we think there's probably a lot more variability than what we've just classified based on 16S RNA. And the structure of these, the structure and ecology of these biofilms are really unexplored, and I think a really interesting topic of study, and something I know people in my lab are still interested in. If you can find out ways to get funding for it, because nobody seems to want to fund that sort of thing anymore, but but a very interesting project, particularly, and you don't need a lot of complex things. That reactor I just passed around, I'll talk about it again in a minute, costs about $1.50 to make. And so you can make 50 or 100 of those with one power source and run an incredibly large number of experiments very inexpensively. So let's talk about that reactor a little bit more and some of the other reactors that we use to try and scale up the system, going from this very small 5 milliliter bottle up to a 1,000-liter reactor test that we did with an MEC. So this reactor, the one I passed around, this is just a bigger shot of it. Doug Call came up with this. Doug is now a professor at North Carolina State University. I once remarked to Doug, I said, there are too many publications in this area. It's like somebody sticks 2 electrodes in a bottle and they think they can get a publication out of it. Whereupon we stuck 2 electrodes in a bottle and got a publication out of it. But it's kind of interesting because these graphite, you can take a piece of graphite. We had them cut up by a machine shop into these little blocks. You drill a hole in them. You take a piece of stainless steel, high non-corrosive stainless steel. You can actually use 2 graphite blocks as well, but then you get confused about which one's the anode and which one's the cathode. And you lose your experiment sometimes because you hook the wrong wire up. And then you can hook just arrays of these up to the same power source. So you can literally run 10, 100, 1,000 of these things off of a single power source. So very, very cheap. They can be autoclaved, and you can do sterile work in these. So very flexible as a platform. You can use bigger bottles. I wouldn't go smaller, but you can use bigger bottles if you want more room for reference electrodes or other things in the system. And that concept of a single chamber microbial electrolysis cell is sort of what's embodied here in this test. This is about 2.5 liters now, where we have brushes and some corrugated stainless steel. But one of the big problems with this is that we would get hydrogen being consumed by other microbes, and so especially methanogens. And so the hydrogen recoveries in these systems were very low, even though they worked fairly well. This is sort of our modified cube reactor. So we have the same reactor I passed around yesterday. It could have a flat anode or a brush anode. But then you put a second chamber onto it with a membrane in the middle, and then you can evolve hydrogen off that electrode, which then bubbles out of the gas into a tube, which is then connected up to a gas bag. So basically, you can set up these reactors to run. You can do this with the little reactors, but we tend to let the hydrogen accumulate in the little reactor because they're very gas tight. And so that pressure buildup isn't a problem in a little bottle. So, for this thing, there's no gas outlet. You release the gas pressure, which can lead to high hydrogen, dissolved hydrogen This one, it's always 1 atmosphere. It releases the pressure, and then we collect the hydrogen and quantify it. So we built upon that concept of a 2-chamber system where now we want to be able to have flow, and particularly across maybe the cathode to strip that hydrogen off the electrode so it doesn't accumulate and repress hydrogen. And then we can have the main body where we have our brush anodes producing the current that will evolve hydrogen on the other side of the membrane. And this is just showing you kind of a cathode chamber and an anode chamber. Some other people have come up with different designs for larger reactors. This is what we call the cassette idea, where the anode is, say, on felt on the outside facing the solution, and then the hydrogen is evolving inside this chamber. And so you can make one of these little cassettes and drop it into a tank. And this was used to treat wastewater. This is a study by Elizabeth Heydrich at the University of Newcastle in the UK. And the recovery of hydrogen was not bad, although their energy balance wasn't that good. So they had to apply a pretty high voltage. This was our reactor, which was 1,000 liters. I'll tell you about these tests in a minute. And you can see this is 24 modules, so 24 anode-cathode pairs, and the wastewater is flowing here, would flow here up through this array of anodes and cathodes. And the anodes are brushes, and the cathodes are stainless steel mesh. So we had money to do this. We thought we would go evaluate some different wastewaters to see where it might make best sense to go to do this. So we looked at 4 different wastewaters: a domestic wastewater, a winery wastewater, dairy wastewater, and a potato chip wastewater. We quantified each of these wastewater in terms of their CODs, the domestic being kind of low, how much power we produce in a microbial fuel cell, and then how much gas or performance we get in a microbial electrolysis cell. And so if you look at, in terms of the flow rates, in terms of gas production per volume of reactor, the scientific analysis was that our best bet was to go to the potato chip factory. However, we're also human beings. And so we decided that maybe the winery would be a lot more fun. So the winery turned out to be nowhere near State College, Pennsylvania, because we don't make good wine in State College, Pennsylvania. But they do make very good wine in Napa, California. And so we decided, why not? We'll do this demonstration in Napa, California. We had a winery there that was very interested in this technology. So we built this reactor and had it shipped up to Napa. And that's where we ran our tests. And without going into all the details, each one of these modules was hooked up to a separate circuit. This is what we predicted we would produce from each module in terms of current. And so you could see, some of these modules did okay, some, close, some lower. The lower numbers can be due to not good electrical connections. We didn't know as much as we know now. Probably would have done the electrical connections a little bit differently. The good news was we got 16 times more energy recovery than the electrical energy we put into the system. The bad news was we didn't get any hydrogen out whatsoever. The electrical energy we put in there did stimulate hydrogen evolution, but it was quickly consumed. That hydrogen was quickly consumed by methanogens. And so really what we were running was the very first sort of electrically assisted anaerobic digester at large scale. But the energy balance was certainly quite favorable. Would it have been as favorable without all the electrodes? We didn't have time to do that experiment. The thing about a winery, and we knew about it, but we went forward with it anyway, is that it's a very seasonal a very seasonal farm, right? You grow the grapes all summer, you harvest them in August, September, you crush them, and then in December, you go home. So, we had all these experiments, and as December got closer and closer, we knew our deadline was coming up. And sure enough, the wastewater strain started dropping. We couldn't do any more experiments, and so everybody had to go home. I will say one of the great aspects of this was if I were to set up a reactor at a wastewater treatment plant treating, say, domestic wastewater or pig wastewater or hog wastewater, I wouldn't get any students that would volunteer. But absolutely everybody in my lab volunteered to go run this reactor in Napa, California. No lack of volunteers, yes. So, you know, our research now has looked at, okay, how do we recover the hydrogen? How do we increase rates? How can we make these systems work better? You know, are there better catalysts than stainless steel? Can we make the reactors more compact? If we're going to have to use membranes, what are the best membranes and things like that? So, you know, I talked yesterday about this module design. This works the same way in terms of scale up with an MEC. It's just that your cathode, instead of being open to air, is now enclosed, and you would capture hydrogen off it. But it would work the same way. It's not quite that cassette design, because the anode does not have to be, the anodes are independent of the cathodes. They're still independent modules. Whereas in the other cassette design, the anode and cathode are bolted together. They're all one module. So the other difference here, too, of course, is in addition to having current and surface area, you also now have to capture the hydrogen that you produce. I'll get to that in 1 second. So a lot of the work I'll discuss was done by a postdoc, Kyung-Yo Kim, who's now at SUNY Albany, using this reactor and looking at trying to increase the rates of hydrogen above 2 liters per liter per day. The catalysts, there's a lot of studies on hydrogen evolution catalysts. And this is a plot by, I forget his first name, Professor Jaramillo at Stanford, showing what's called the volcano plot, because it crescendos here in platinum and palladium and ruthenium and lithium disulfide and nickel. So nickel is indicated to be a good hydrogen catalyst in terms of being expensive and not being a precious metal. And so there's a lot of interest in using cathodes that have hydrogen. And in fact, stainless steel has, catalysts that have nickel, and stainless steel can have a high nickel content. So we looked at the architecture of the cathode as well as the material of the cathode. So just looking at stainless steel first, in terms of, is it a mesh? Is it a fiber felt? Is it a wool? Or maybe a brush design like this? How much does the physical architecture of that cathode impact performance? Because the flow can be coming up through the stainless steel, or maybe across these, or through the brushes, and so forth. And so comparison of this basically said that the stainless steel wool is going to work well, but it's also got a much higher effective surface area than any of these other materials. We looked at flow rate. This is just showing, this happens to be the mesh. But overall, you could see that when you had a little bit of flow over the surface, you got, a little bit of flow over the surface. These are different flow rates here. That just a little bit of flow, made a difference. Because this is the green up here. And these are over potentials. So there's a little bit going on there, but the flow rate wasn't nearly as important as the material. And here you can see this stainless steel wool. And as a reference point, we show a platinum catalyst. And so, this platinum is always going to do better than stainless steel or nickel or anything else. But this stainless steel wool did pretty well. So then we used that stainless steel wool in microbial electrolysis cell tests with these brush anodes and the cathodes. And we used a synthetic fermentation effluent. We tried to represent the kind of cellulosic fermentation effluent that you would get from the reactors being tested at the National Renewable Energy Lab. And in these systems, with flow in this reactor, we did see a much more pronounced impact of flow rate in the system. That's true for platinum, for mesh, for all these other ones. And so the stainless steel wool actually over a cycle did about as well as the platinum. The platinum worked better at the beginning, but as substrate went away, the rate slowed down. And so a combination of biological and chemical reactions meant that actually the stainless steel wool over the cycle did about as well as platinum. I mentioned the nickel. People have done experiments with nickel foam cathodes. Some of the highest rates ever achieved in a microbial electrolysis cell for hydrogen evolution was done with a nickel foam cathode. But of course, if the cathode corrodes, you'd have to replace it. If it plugs, you'd have to replace it. So one of our ideas recently was to look at adsorbing on nickel into activated carbon, because activated carbon has a huge surface area. If you could absorb the nickel into the carbon, then you might have even more effective surface area than you would have with stainless steel or stainless steel wool. And you would have the benefit of the nickel catalyst. And so you can see here, this is the content, the 2.2, 4.4, 8.8. That is the nickel content, and this is your carbon oxygen. So this is essentially your carbon underlayer, which is blue. And as you put more and more nickel on, you get this sort of more yellowish structure. And we did some abiotic tests. These are just chronoamperometry scans where we set the potential and see what the current density is that evolves under steady conditions. And you can see that some of these nickel ones did pretty good. This nickel 8.8 is the green one here. It depends what current range you're in terms of which one's doing better, which we don't fully understand. But the microbial electrolysis cells tend to operate up in this range. So, a lot of times we look down in the high current density range to see how the catalyst looks, but actually we're operating way up in the top part of that where there's a lot more overlap. So this is a comparison of nickel foam versus activated carbon with nickel. And you can see they perform pretty similarly. This is the green ones, the average current density. This is the hydrogen production rate over a cycle. So they work about the same. But of course, you could say, well, they cost about the same too. So what's your real advantage here in using this nickel on the activated carbon? And I have to say, well, I don't know. They look pretty close. But the nice thing about the activated carbon is it will absorb more nickel. And so actually we absorbed, after some tests, we intentionally acid washed the nickel, the activated carbon nickel. We tried to get off all the nickel, and then we reabsorbed some more nickel. So as a way to regenerate the cathode, whereas you'd have to, say, replace the nickel foam. And actually, when we did that, that reabsorption step made it work better than it worked previously. So there's still probably ways to improve this nickel absorption test, and it's kind of interesting in terms of thinking of other things you could absorb onto activated carbon to make this work better. So this is something that remains of interest, but we truly don't know which one's the best right now. So, in terms of MECs, where have we really come? I talked about yesterday microbial fuel cells. Where have we kind of come in terms of production rates? In small reactors, these 5 mil reactors here, we have 86 square meters per cubic meter of electrode surface, getting up to as high as almost 200 amps per cubic meter, which translates to about 1.7 cubic meters of hydrogen per cubic meter per day. I love using the word cubic meter when I'm talking about 5 milliliters. So really, I should be saying 1.7 milliliters of hydrogen per milliliter per day. Larger reactors, 64. And then I said, okay, what's the best we could possibly do? Okay, so maybe if we got up to, say, 50 amps per square meter, which we haven't gotten to, and we were able to put in 50 square meters per cubic meter, we would recover about 2.2 kilograms of hydrogen per day per cubic meter. to compare that to, say, a water electrolyzer, people have talked about using home water electrolyzers to produce hydrogen for vehicles in your home. And these small ones produce about 1 to 2 kilograms of hydrogen per day. So our reactors are bigger than those, but gives you a sense of what a small electrolyzer will do versus something like this. So conclusions, single chamber reactors are more efficient, but they you often lose that hydrogen to methane. The hydrogen methanogens are quite fond of methane, hydrogenotropic methanogens. Catalysts, there's a variety of catalysts possible. Stainless steel wool and nickel are really 2 promising ones that are alternatives to platinum. And the upper rates, so far we've only gotten typically up to 2, maybe 3 or 4 liters of hydrogen per liter of reactor per day. The highest rate I've ever seen is 50. But again, that was in a really compact reactor that if you tried to put a wastewater in it would flood. So that brings us to the third part of this, which is to talk about the biocathodes and things that you can produce using the electricity, say, produced by the anode. Or, as I also said, you could split water and do the same thing on the cathode. But the idea here is that these microbes can facilitate the reduction of a lot of things, oxygen, nitrate, but also CO2. And so what can we do with that? So I won't talk about dissolved oxygen and nitrate. Also, I will not talk about metals reduction. This is also used as a way to get metals out of solution in metal-laden wastewaters, particularly copper. It seems to work quite well with. But this is our conventional textbook description of fermentation and methanogenesis. You say that there are microbes that ferment substrates like glucose into hydrogen and acetate, just like yeast can ferment glucose into ethanol. These bacteria can ferment substrates into hydrogen. And then that hydrogen, these microbes grow quite fast if you give them soluble glucose. And the electron flow through a methanogen is actually quite fast as well. These don't grow fast, but they process a lot of hydrogen, and they're quite optimized for the rate at which they can process hydrogen through their electron transport chains. So what you have is a really fast production of hydrogen, a really fast processing of hydrogen to methane, with a very slow transport step in between. You have to dissolve the hydrogen, has to diffuse over to the methanogen, has to be taken up by the methanogen. That seems to be counter to nature optimizing systems. And so it makes much more sense to have these microorganisms directly coupled. And so that was our hypothesis going into this, that actually these electrotrophs, electrogens as in generate, electrotrophs, will find ways to interact with electrogens. And this has been validated now with a number of different microorganisms, but this is the early study with methanogens. And so what we did is we grew cultures splitting water, so we had no complicating microbiology on the anode. We just focused on the cathode. And we found that we could develop a culture of methanogens that was dominated by Methanobacterium palustri. And what you can see here is this is the amount of methane coming off the cathode over time. And based on the abiotic rate under the same applied potentials, this is the rate of abiotic hydrogen that would come off that electrode. So clearly, the presence of these methanogens was catalyzing the release of electrons somehow, and thus enabling a high rate of methane production. We also did it with a stock culture from ATCC of Methanobacterium palustri. We also saw a higher rate than the abiotic rate, but the rate by the stock culture was nowhere near as high as the rate by our mixed culture dominated by Methanobacterium palustri. So we talked about bogs and wastewater treatment plants and using that as anodes. We also did the same thing with the cathodes. We looked at enriching microorganisms on the cathodes in these MECs. And what we found was here, this is the community analysis of the seed versus what we found on the anode and the cathode in solution. And what you find on the cathode is basically this black. This is black, and then this is Methanobacter. This is Methanobacterium. So when we inoculated these and we ran them on the cathode, we got predominantly Methanobacterium species, genera on the cathode. And we could have a seed that was quite diverse relative to the methanogens, but after cultivating them in these reactors, it was almost all this one genus. We also looked at different materials and how that might impact the growth of methanogens. And this is work by Mike Segrett, who looked at making cathodes of all these different materials, and then as well as one of platinum. Now, platinum's a great hydrogen catalyst, right? So we knew that when we ran the experiment, this would evolve a lot of hydrogen. And these other ones differ. Steel and nickel are okay, right? They're good catalysts in their own right, but we had some other things we weren't too sure about. We also had brushes and just graphites, things which are terrible hydrogen evolution catalysts. And you look at the gas production rates, and the methane that's produced is converted to hydrogen. So we divide it by 4. The hydrogen is converted to methane. So we take the current and translate that rate into methane equivalents. And so on the basis of likely hydrogen bubbling off here and being converted to methane, these are the methane rates that we're getting. And you can see that the methane rates from some of these other materials approach those methane rates of even the platinum. So it's clear that these microorganisms are finding ways to evolve methane very fast and almost as well as platinum in some cases. And when you look at the microbes that evolve on these electrodes, you see that no matter what it was, except for this one, no matter what it was, it's almost all methanobacterium. This is open circuit controls. These are our active cathodes, and this is our platinum. And the platinum one has very little of this methanobacterium on it. It's got mostly Methanobrevibacter. And so we say when hydrogen's evolved, yes, you're going to see this Methanobrevibacter, but if it's a poor catalyst or not as good as platinum and likely therefore to be evolving a lot of hydrogen, we end up with Methanobacterium. So we don't really understand how these interactions are occurring, and I could talk for another hour on some of the studies that have been done, but I won't do that to you. I'll just leave you with some concepts here about this process. One is we think these electrogens either produce conductive pili or they have surface active cytochromes on their, outside the cell, which can transfer electrons. And then you have the electrotrope, which is somehow going to interact with these pili or surfaces of the microbes. And we have to wonder, in nature, is this a very specific relationship, right? And so, is this like a plug and socket? You talk about enzymes and substrates, right? Is this like because we're talking about electrons, is the analogy of a plug and socket we think makes good sense. Is it like a plug and socket relationship? But, if you've ever traveled outside of Mexico or the US, not all plugs look the same. And so the question is, how specific is that pili for that plug, right? That's the UK, that's Europe. Or maybe the electrotropes have sort of figured this out, and they go to universal adapters, and they'll accept any pili, no matter what it looks like. Or maybe everything's just, it all fits together. There's no specificity. So there's probably some really interesting things with certain cytochromes, with certain pili, with certain interactions. Again, the ecology of these systems can be quite fascinating. And what we've seen in recent years is people looking at this more and more. And this is just sort of a survey. OK, is it abiotic hydrogen? Is it a bacterium that releases, say, hydrogen or something else from an electrode to a methanogen. Is that what's happening on the cathode? And we find very little evidence for those mechanisms to be occurring with this with the studies we've seen where there's little hydrogen. But it could be abiotic. Well, we don't think it's abiotic because those are very low. Is it some sort of hydrogenase? Is it a catalyst that's made by the methanogen? Or maybe methanogens produce their own nanowires. We think, really, as we've gone through this, that it could be that one. But most recent data suggests it's probably not hydrogenases. That leaves us with other. And I categorize that as the fairy dust explanation. It's like we don't know what it is, but we sprinkle a little fairy dust on it, and it works. So there's something going on here which we don't really understand, which should be investigated. But we find it interesting. There have been people who have been saying, when we look at these aggregates from anaerobic digesters that have methanogens and organic matter, they turn out to be highly electrically conductive. Why are these aggregates electrically conductive if this sort of thing isn't going on? There must be some sort of electron exchange going on, because surely it can't be by chance that these granules are electrically conductive. People have also looked at promoting direct interspecies electron transfer by putting conductive materials like magnetite or activated carbon. In my opinion, that's very circumstantial because you're increasing the surface area available for the microbes, and whenever you increase surface area, you can increase rates. So I'm not sure how well that will play out, but there's a lot of interesting things occurring there as well. So, what have we achieved in terms of electromethanogenesis, the conversion of electrical current into methane? Well, in small systems, rates of 1 to 2 grams per cubic meter per day. In larger reactors, several times that in terms of volumetric current densities. If we assume optimal rates maybe of 50 amps per square meter and 50 square meters per cubic meter, we might be getting up to something like 4.5 kilograms of methane per day. That's just because you have the carbon in with the hydrogen, so that's just a conversion of hydrogen and fixing CO2 to make it methane. Comparison, an anaerobic digester, a really good one, can produce about 10 to 20 kilograms of methane per day. So we're not at the rates that anaerobic digesters could achieve, but likely because these are surface-limited reactions. So, but anaerobic digesters have their own advantages and disadvantages. And that's a subject for another day as well. OK, that leaves me with my last subject, which is just a few minutes long. Again, the idea is we can make hydrogen, which I would argue is the best possible thing to make. It's versatile. It's highly energetic. You don't need CO2. And you can use it for so many things. So much of the hydrogen we make today is used for what? Not cars. Ammonia. Haber-Bosch. Convert fossil fuels into hydrogen and use that hydrogen and nitrogen in Haber-Bosch to make fertilizer. About a third to half of all the hydrogen made is made to make fertilizer. That's a conversation for another lecture, another day. Okay? I will also say that we could power most of our light-duty vehicles by hydrogen using the amount of hydrogen that is made at refineries to produce gasoline. All that hydrogen using to crack alkanes and to make low alkane hydrocarbons. If you took that hydrogen, you could just run these fuel cell cars on that hydrogen. They get much better gas mileage per kilogram of hydrogen than cars do per gasoline equivalent. Oh, OK, I got off on a tangent. So, different things you can make here. I've talked about hydrogen, methane. Let's think about acetate. So acetic acid production. What people have found out is if they make acetate and they don't evolve hydrogen, it doesn't happen very fast. The direct electron transfer to acetogens, microorganisms capable of fixing CO2 and making acetic acid, it's quite slow. And so a lot of the studies that have been done are actually done at current densities where hydrogen must be furiously coming off the cathode. And they detect hydrogen, and that's well known. So, what's the prospect for microbial electrosynthesis? Here are two different studies, one done by Jourdain in Australia, this one done by Cornel Rabai's group in Ghent. They achieved 0.5 kilogram per day per cubic meter of acetate, about the same thing achieved by Gilderman with a totally different reactor design. Let's say we can get up to 50 square meters per cubic meter of packing of electrodes. And now I'm going to double what I said before. Let's say we can get up to 100 amps per square meter because we're bubbling off hydrogen. So that's 5,000 amps per cubic meter, or about 34 kilograms of acetate per day per cubic meter. So that's what we could achieve, I think, best case scenario. But look where we are. We're down to the half. So you could say, well, this is good. This is a great opportunity for research. We can get, if we can increase these rates, that's good. But I want to put another thought in your mind as a contrast, and that is to make hydrogen however you want and then gas sparge that hydrogen into a reactor, because that's mostly what we're doing in microbial electrosynthesis. We're mostly just driving hydrogen evolution. And if we're not doing that, the rates are low. That's okay if the rates are low and it becomes economical, but most people are just making a lot of hydrogen. So the question is, from a chemical engineering point, how do you convert those two, how do you put these two things on the same basis, right? What do chemical engineers use to compare these reactors? Anybody know? Come on, students. Just if I were to bubble hydrogen or oxygen or anything into water, what coefficient would I use to describe the rate of mass transfer? The mass transfer coefficient, KLA, right? So for this, we use KLAs, but for this, we use amps per cubic meter. So you got to put those on a similar footing. And everybody always says it's bad to put up equations, so I'm not going to put up equations. I'm just going to tell you the results. If I compare current densities, it goes to electrons, coulombs, rates of mass transfer, all this stuff. If you put them on the same basis, an MEC, this optimal MEC, could produce a KLA equivalent of about 63 per hour. Stirred reactor can average around 300. Some other types of systems based on baffling and so forth, a membrane reactor and a microbubble reactor up around 1,000 to 2,000. So just in terms of how much hydrogen can we get into the system, I don't believe the MEC or the MES is going to compete with this on a rate based on solely on hydrogen production. So that's not to say that MES isn't good, but it is to say that there better be an advantage that's not strictly hydrogen production rates. And this reactor by the group in Belgium uses the current generation also to do separation, chemical in situ chemical separation, which is good because you got to get the acetate out of there somehow. And they confine it into this area at a low pH, and then they strip it out later. So, if you want to look at current densities instead of KLAs, we can say that we're looking at sort of 50 amps per square meter, and packing densities, that's current densities of about 0.5 to maybe 5 milliamps per centimeter squared. I'm going to those units because a water electrolyzer does about 200 to 400 milliamps per centimeter squared. So if you're splitting water, this is the kind of rates you can do. If you're doing this in an MEC, those are the kind of rates you can do. If you have just electrodes that have a certain spacing and it's not very conductive liquid, just the ohmic losses alone will limit you to about 10 milliamps per centimeter squared. and this is the best KLA for gas infusion. So, you know, again, looking at rates and current rates versus mass transfer rates and things, I think we have to keep that in perspective. We also have to realize that if we're going to use hydrogen and CO2, that we have to sparge CO2 into this system. So it's an open field. I think it's really interesting about how these microbes can interact with this and the products they can produce. But we can't lose sight of other reactors as alternatives as well. So that's the third and final part of my talk, using electrotrophs, and particularly methanobacterium and methanogens for electromethanogenesis, the production of methane from an electrode, which can or cannot be, which may or may not be catalyzed by hydrogen evolution. We can get also other products. Acetic acid is one of them. People have gotten other products by sort of upscaling acetic acid to a higher number of carbon groups. But we're going to have advantages just more than effective mass transfer from an electrode into solution, because the rates are going to be low to these electrochemical systems. And I'll finish this up by saying thank my sponsors and my collaborators, the Ghent researchers who I've worked with on this quite a bit, my colleagues in China, support through the National Science Foundation, and as always, KAUST for their generous support for me. And that's my research group, which decided to have our picture indoors instead of outdoors. This is our usual desire, but this was during the polar vortex. So it was 0 degrees Fahrenheit or minus, what is that centigrade? Minus 20 or 30 centigrade, so we weren't going outside for this picture. So we stayed inside for this one. And of course, a plug for my favorite journal. I always like to end with that and leave that up there while I talk about other things. So thank you, and if you have questions, I'd be happy to answer those.
Okay, the hall is open for questions and comments. So go ahead.
That's so they can get it on the
Okay,
report.
sorry.
Yeah.
When you're reporting the results in terms of the current, are you referring to limiting current densities, right? In order to have the conversion between the mass transfer coefficient to currents.
Yeah, what I'm doing is I'm saying there's a certain amount of electrons being transferred, therefore a certain amount of coulombs. Those coulombs go right into the half reaction for whatever we're producing. So many electrons mean so many units of product. So whether you're using an electrode to make hydrogen at a certain current density, or whether you're sparging hydrogen in with a KLA of whatever it is per hour times the C0 C saturation, that's the rate you can get. And so you can, just on a basis of mass of hydrogen for both those cases, you can put them on a KLA equivalent. That's how you do it. I have the equations if you want to see them.
Another question? Comment? Okay.
Let's see. Oh, no, maybe I don't. There's KLA, and there's the units. I'll leave those up for you. Yes? Yes?
How can you differentiate methanogenesis, diffusion through membranes from electromethanogenesis?
How can you make sure I understand this? How can you?
Differentiate between electromethanogenesis
Yeah.
or diffusion of methane through the membrane.
So are you saying, so for example, let's say I have an anode, I'm making methane in the anode, and it's diffusing through the membrane to go into the cathode.
And you detect
Yeah. The only way we really know is when we have a good sterile catholyte. If that's true, then only the methane that we would detect in there would be the methane that leaks through the membrane. And so typically, if it's an ion exchange membrane, we see very little methane transport through the membrane. However, there's probably not there. There is more hydrogen diffusion back because, you know, hydrogen's this size, methane's this size, and so it's easy for that hydrogen to diffuse back. So the loss tends to go of hydrogen into the anode chamber, not so much methane. Yep. Yep.
Okay, another question? Yes.
Please.
Well, probably it's a silly question.
No, I love silly questions.
Well, how? What? Hello,
It's
hello.
just for the recording.
Okay.
It's not gonna
How
work for the
nickel absorb the activated carbon?
How what?
How nickel absorb the activated carbon?
Oh, how much nickel absorbs into the activated carbon? So in the numbers that I gave you, it was 2.2, 4.4, and 8.8. That was a certain mass of nickel absorbed per mass of activated carbon. And I think it was milligrams per gram. So it's not much. But, and what we really don't know is how active surface area-wise that nickel is on the surface. So, and we don't know what the upper limit for that absorption is, right? You know, like from a Langmuir or absorption isotherm, it can keep going up and up. So we haven't really explored what the upper limits are. But we do know if we start to absorb too much that it's going to desorb, right? Because reversible absorption, desorption. So at low levels, it seems to stay on there pretty well. But if we get up to the higher ones, we do start to see a little bit of desorption. And of course, we don't want that nickel going into the water. Yeah.
Also,
Not a silly question.
adding to the question of him is, what kind of process did you use to conduct the absorption of the nickel into the?
It was
Activated
really,
carbon.
so the method to absorb it was very, just a nickel salt in the water and shake it for 24 to 48 hours. And the pH is important. Low pH, you can get, you know, in acid solutions, the nickel will readily come off. So the pH has to be around 7, you know, it has to stay around above 6 or 7. In that range or higher is fine, but below that, you can start to see dissolution. So an acid wash pulls almost all the, you know, pH 2 will pull off almost all the nickel.
Okay, another question. Okay.
Oh,
right.
yep.
Have you done any metagenomic analysis to these microbial communities to know sort of the kind of interactions that it's been made by all of these microbes?
No. Great interest in that. I mean, really, that's where the field is going, right? To know what's being expressed, what's being upregulated and downregulated, what those things do. I think there are people with, there's the movie Taken, where the guy says, I have a very special skill set, right? Well, unfortunately, in this case, I don't have that skill set. So that I leave that to other researchers. But I think that for people with that skill set, it's a fascinating problem, a fascinating thing to study. And so I really look forward to seeing what they find out. Some of the other things that are going on are people testing what they think is going on by translating genes from, say, Geobacter to put their pilus, the Geobacter pilus, into another microbe and see if it changes its performance, or to take the pilus from another microbe and put it in Geobacter and see if it can work in Geobacter. So these kinds of switching of you know, functionality between these microbes and to looking at what's expressed are really fascinating, I think, frontiers of research in this area.
Okay. Before thinking another question, I have one question. Indeed, several questions. The first one is, you show us that you apply a voltage of 0.9 volts to conduct the hydrogen evolution reaction. And I want to be sure if this voltage is possible to use, because the theory mentioned that to conduct this reaction, we need approximately 1.2 volts. And the question is, how did you achieve this reaction using only 0.9 volts?
Yep. So when we split water, we use 0.8 volts at the anode, and we require about 215 volts at the cathode. So the total input energy for two thermodynamically unfavorable reactions is about 1.2 volts in theory and about 1.8 volts in practice. We have to, because of ohmic losses and overpotentials, we have to apply a greater voltage. In our system, the anode reaction is favorable. We actually derive a favorable minus 0.3 volts, almost minus 0.3 volts at the anode. So instead of needing 0.8 volts, we now gain 0.3 volts. So if you look at the cathode reaction, you could say, well, it's 0.415 at the cathode. So we make minus 0.3, we need 0.415 at the cathode, therefore we need 0.115 theoretically. And in practice, we don't see anything until we get up to about 0.2 to 0.3 volts. So it's really that favorable oxidation of organic matter at the anode which makes this so much more thermodynamically favorable.
Another question? No? Okay, let's another
Yeah.
short. Also, in the results that you present in terms of the hydrogen production, you mentioned that, of course, when the methanogenesis occurs, you are losing hydrogen, okay? And I would like to ask you, you didn't control the methanogenesis in such a way that you can increase the hydrogen production.
Yes.
And if you do that, how did you control the methanogenesis?
We've tried a lot of different ways to control methanogenesis. None of them particularly successfully in terms of a practical aspect. There are several known inhibitors of methanogens, and those can be used in the electrolyte to inhibit methanogenesis. But by and large, these chemicals are expensive and can be toxic at higher concentrations. And so we've used them to inhibit methanogenesis, but in a larger scale system, it would never be practical. Was there something else I was going to say on that? Yeah, the best way to do it, of course, is not to have any to start with. So if you have an industrial process where you could control the community to keep it relatively sterile, then that wouldn't be an issue. So, yeah.
No other question? Well, I have a
one back there.
Good.
Could you take advantage of the direct electron transfer into the cell to produce methane?
So, so you're saying, can the electrons go directly from the electrode right into the methanogen? Yeah, I think that's what we believe is happening with this Methanobacterium palustri, because they are enabling a high current density, but it's not clear that they're doing anything that necessarily catalyzes hydrogen release. So we reason it's direct electron transfer. Some material I didn't have time to cover by Alfred Sporman at Stanford University, who used Methanococcus maripolutus, which we do not find in our system, but which is a well-studied methanogen. They knocked out all the hydrogenases, and it wasn't a hydrogenase for that microbe. And then they took the solution and filtered out the methanogens, and they still saw a high current density. In other words, that methanogen actually does excrete a fumarate hydrogenase, which catalyzes the evolution of the hydrogen from the electrode. So there are species that are doing this catalytically evolved hydrogen, But we have no evidence yet that methanobacterium does anything like that. So we think it's direct electron transfer.
Okay, I have a question, and it's some kind of a thinking, the possibility to use hybrid systems, I mean microbial fuel cells and microbial electrolysis cells, in such a way that we can use the electricity that we can produce from microbial fuel cells to supply this electricity to the microbial electrolysis cells. How do you think about this possibility? In such a way, for instance, you show us that with 0.9 volts, we can conduct the hydrogen reaction evolution. And if we use, I don't know, 0.5 volts or even more from microbial fuel cells, probably we can set a system that's more efficient in terms of energy balance.
The answer is yes. We can link together microbial fuel cells and microbial electrolysis cells. What we do find, though, is we might need 2 or 3 microbial fuel cells per microbial electrolysis cell, because the voltage produced by a single MFC is about 0.3 or 0.4 volts, only about half what we would want for the MEC. Alternatively, we can just run a bunch of MFCs and charging capacitors and then have the electrical circuits take care of the voltage and current the other way. So yeah, it's quite feasible to technically have these MFC power MECs, provide the power that we need in those MECs. It's also, I'll mention research done here, which also says that if you have an MEC and you have a very acidic catholyte, you don't need to add voltage, because the cathodic reaction becomes so favorable that it will drive the reaction without any power addition whatsoever. But you have to have a very acidic catholyte available. It costs money to make acid to put into the catholyte. So in the right circumstances, you might not need any power at all.
Okay. Probably the last question. Yeah.
What is the half-life of the cells before you need to change?
So I assume you're referring to just a microbial electrolysis cell. That's a far more complicated question than you know. And it has to do with the fact that when we run these cells at very low current densities, they actually run for a very long time. When we run them at high current densities, the bacteria, to anthropomorphize, to make them sound like humans, they get tired and they quit. So we've had reactors that are running really, really well, and all of a sudden they just quit. The current density starts falling. We don't know why. We don't know if it's a bacteriophage invasion. We don't know if it's literally the cellular components are breaking down. We're missing trace nutrients. There's an imbalance there. So at low current densities, they run pretty well for a pretty long time. I think the weakest link in the whole system in the MEC is the membrane, because the membrane can foul. So that could be the weak link. Complicated question, yes.
Okay, with this, we close this session, this morning question. So we will take a 10 minutes break.
Sounds good.
And return for the last workshop. Okay? So,
all right.
Thank you, Professor.
Thank you.
Thank you very much.
So, you see the calculations?